Okay, so I want to give you a quick run through on um, DNA libraries and screening techniques. I want to bring to your attention the most important things that you should take home from the notes on construction of libraries and screening, the ones that I alluded to. The first thing is the definition. You need to understand what your DNA library is, and it's a collection of cloned DNA fragments. So, what that simply means is that just like a library where you go to look for a book, the book is your plasmids, and the pages are the DNA fragments in there. I think that's a good way to look at it. So each plasmid represents a book. Let's put it that way. There are two types of DNA libraries. There's genomic DNA libraries where the whole genome is represented. So this library means that if you have a genome of, let's say, Drosophila or E. coli, that whole genome is cut up into little bits and it's cloned into plasmids. Whereas a cDNA library is only expressed genes. So all the genes that are expressed are what is represented in the library, not all the untranslated stuff. So those two types of libraries are used in different ways. So how do you construct a genomic DNA library? It follows these steps. You isolate the genomic DNA from your organism, whether it's Arabidopsis or Musculus, whatever you've chosen. Then you fragment the DNA, you ligate it into cloning vectors, such as PUC18, that you're used to, and then you introduce it into your host cell. So that would be your heterologous expression. Then you select, let's go back here. You ligate it into cloning vectors, and then you introduce it into the host cells. So that would be your E. coli. So now you'd have your bits of DNA in plasmids which are in your host cell, such as E. coli. Then you select and look for clonal growth, and you use antibiotics or different mechanisms to find your plasmids that contain insert DNA. And you have to store and maintain your library, because if you don't do that, you lose it. So normally we do this at minus 80 degrees. That's where we store our libraries. And our libraries are usually stored in microtiter plates, which have 96 wells. So a whole library, let's say, of Pseudomonas, for instance, a cDNA library of Pseudomonas might be in a library that has maybe 10 or 12 microtiter plates. So that's the kind of dimensions you're looking at. Then when it comes to DNA isolation, that's the first step. How do you isolate the DNA? You usually use some kind of lysis method, and you remove the protein, which you know well now, and you can fragment the DNA with mechanical shearing or with restriction enzymes. So with mechanical shearing, you're using a physical mechanism. With restriction enzyme, you're using a molecular mechanism. Then you ligate it into your vectors, and you know the plasmids that you've dealt with so far. But there's other vectors you can use, such as lambda-phage or BACs or YACs. And BACs are bacterial artificial chromosomes, and YACs are yeast artificial chromosomes. Then you have to remember about the compatible ends for insertion. So you have to restrict with the right combination of restriction enzymes so that they can ligate nicely. And you need to understand the role of DNA ligase in this process. So once this is done, you have your circular plasmid, which has your piece of the library. Then you need to select. So you transform into your host cells, such as E. coli, and you use antibiotic resistance that's present on your plasmid to be able to select the plasmids that contain your insert. So each colony that you see on your library is a unique DNA insert. Excuse me. So the whole purpose of screening is to identify the clones with the gene or the DNA of interest. So after you've made the library, it's useless if you can't screen it to look for the things that you want. So you've got different mechanisms of screening. And here we've got nucleic acid hybridization. In this case, you're actually using the sequence of the DNA. You make a complementary probe, which has something attached to it that is either fluorescent or can be seen, and you end up having to hybridize your pieces of nucleic acid to your library, and that piece of complementary probe will stick to the sequence within your library which is complementary, and then it will make that particular colony shine and or fluoresce, and so you'll be able to see what it is, which one is actually carrying the gene of interest. So how you do the procedure, you lift the colony, which has the genes of interest. And the way this happens is you have your whole library and you're able to clone that library onto separate plates, and you introduce the probe to all of your library so that hybridization can happen. Then use a detection mechanism which detects the fluorescence from your probe. So the fluorescence will only come from when this complementary probe has landed on a piece of DNA, which is the target one. It's complementary. And so then the probe will fluoresce, and you know exactly which colony has your gene of interest. And there's different types of hybridization. You can have colony hybridization or plaque hybridization when you're using lambda or phage type of probes. Like here, where is it? Here. If you're using lambda phage, you're going to be using plaques. If you're using the normal method, then you're going to just look for colonies. Then you can also use screening using PCR, and this one amplifies target regions from your clones. It's very quick, but it's a lot of throughput. So you have to do a PCR for every single colony that you generate from your library. And obviously, when the right target is found, then you find a band on your PCR reaction, and then you know that that's the right one. You can also use immunological assays, and this, you use antibodies to detect the protein products. So this is only applicable to libraries where you have expression. So this isn't your genomic DNA library, but you want a library that is an expression library, because this is antibody, so it's protein to protein. So this is a bit different from the other two methods that we've talked about. You can also do functional screening, where you actually look for a specific function. So something like lipase activity or cellulase activity. So you look for the activity of an enzyme, and in the case of phenol red, that is an indicator that we use. If you have the activity of a lipase, for instance, it might make a mark in your library. So this, I'll show you some examples of what functional screens look like. But basically, they're phenotype-driven selection. So you're having a library which is on your plates, and you'll see something. If there's activity, the enzyme is working, then you'll see something around your colony, whether it's a zone of clearance, or it's a color change, or something. For instance, blue-white screening, X-Gal screening, that is a phenotype-driven selection, and we've talked about that in the past. So, quick summary, you've got different ways of screening. You can do hybridization, where you use DNA or RNA sequence, and that hybridizes to the complementary sequence in your library, but you need a labeled nucleic acid probe. That is the one which fluoresces and gives you insight to where it's stuck to the complementary sequence in the library and makes you find the correct colony. You can have PCR screening, where you're using a DNA sequence, and this can work on any library. It's fast and it's very specific. You can use immunological screening, where you need a protein product, and this only works on expression libraries, and you need an antibody, and it's protein-protein interaction. It's antibody-based detection. And you can also have a functional analysis, where you have protein function. You're looking for the ability of a colony to do something that its friends are not doing. So you're looking specifically for protein function, and this is where you have expression libraries, and it's called functional screening. You're looking for a specific phenotype. Yes, so out of all these, hybridization is the most widely used. And it's up to you which screening method you're going to use, and it's based on what your library is and what's your goal. So screening isolates from your library allows you to get the clone of interest, which you'll use for your further work. And you must always be up to date with online cloning protocols to understand how you actually do this in the lab, and standard molecular biology texts, which go into this in much more detail. So for you, you must understand all the parts of this table and understand why, what are the differences between each of these screening methods. And that's basically it about screening your DNA library.